first of all, I would like to thank Professor Nova for the introduction and inviting me to this seminar. And also, I would like to thank you all for attending my talk. The title for my talk today is RB efficiency covers ATM signaling to DNA methylation profile indispensable for C-cell carcinogenesis. At the beginning, let me briefly introduce RB tumor suppressor protein. RB tumor suppressor protein is a multi-domain protein. This protein carrying binding sites for multiple proteins, including viral oncogenes, transcription factors E to F, chromatin modifiers containing LX, CXE motifs, and tissue-specific differentiation polypeptides. RB proteins or RB gene mutations or deletion are very rarely detected in cancer. However, RB proteins is mainly regulated by post-transcriptional regulation, specifically phosphorylation. And phosphorylated proteins are expanded and its binding sites are exposed for interacting proteins. However, phosphorylation of RB induce conformational changes and folding of the proteins that mask the binding sites of the proteins and bringing the proteins biologically inactive. What is the function of RB proteins? RB or retinoblastoma protein or retinoblastoma tumor suppressor gene was the first tumor suppressor identified 26 years ago. And it regulates several important cellular functions, including cell cycle, transcription, differentiation, genome stabilities. And recently, we showed the novel function of RB in regulation of DNA damage response, cellular senescence, and today I'm going to talk about another novel function of RB in regulation of DNA methylation. DNA methylation occurs at two steps. First step is initiation of the DNA methylation code. Shortly before implantation of fertilized egg, all the methylation code are erased, and the cells have to establish their own DNA methylation profile after implantation in a process called DNA or de novo DNA methylation. De novo DNA methylation is carried out by DNA methyltransferase 3A and 3B. And the second step of DNA methylation is the maintenance of DNA methylation, because the cells has to maintain or to copy their DNA methylation code during proliferation or DNA replication. So DNMT1 or DNA methyltransferase 1 is an enzyme or methyltransferase has a high specificity to bind to hemimethylated CBG dinucleotides and not to unmethylated CBG. And this gives the specificity of DNMT1 in copying the DNA methylation from the parental strands to the newly synthesized DNA strand during DNA replication. Importantly, when we see the protein structure of the mammalian DNA methyltransferase, DNMT1, 2, 3A, 3B, and 3L, the C-terminal catalytic domain is very well conserved, except for a newly identified sequence in the DNMT1 called target recognition domain. This gives the specificity for DNMT1 to bind to hemimethylated dinucleotide, CBG dinucleotides. DNMT2 has no enzymatic activity, but it also share the function with de novo methylation by docking DNMT3A into the genome. DNMT3L is a well-known sensor, it binds to internal domain of H3, histone H3 when lysine 4 is not modified. So this DNA methyltransferase is considered as a sensor for lysine 4 methylation or modification. Also, I would like to give a brief introduction about ataxia telangiectasia mutated or ATM signal related to my talk today. ATM exists in dimers which in active, functionally inactive. When sensing DNA damage, ATM become activated by monomerization and autophosphorylation, and it binds to NBS1 and activate hundreds of downstream effectors to activate the cell cycle checkpoints and initiate double-strand break repairs. Importantly, recently, ATM has been reported to respond to chromatin modification in a similar way to the DNA damage and initiating signals to maintain the genome stability and cell survival. One example of this chromatin modification is histone H3 K9 trimethylation. This is a very well-known mark identified by the acetyltransferase TIP60. TIP60 has a chromodomain, and this chromodomain identifies this histone mark and bind to H3 K9 trimethylation and become activated. And when this TIP60 acetyltransferase activated, it acetylates ATM, and ATM acetylation further promotes autophosphorylation or activation of ATM. So there is relevance here for ATM to interact or respond to chromatin modification. Our working model or experimental model, we are working on RB heterozygous mice. RB heterozygous mice generate tumors in various organs, including C cell of the thyroid glands. C cells are very few cells located in between the thyroid follicles, and they are responsible for production of calcium hormone. So as a consequence of loss of the remaining normal allele of RB, somatic loss of the remaining normal allele of RB, these mice generate C cell adenoma. And others diagnose it as a pre-malignant tumor in the C cells. Additionally, we recently found that additional loss of NRAS, ENC4A, or histone methyltransferase SCOV39H1 convert this adenoma to metastatic adenocarcinoma. And we characterized the underlying mechanism for this malignant conversion from adenoma to adenocarcinoma, and found that RB regulates a bunch of protein isoprenylation genes. And this isoprenylation is an initial and rate-limiting step in RAS protein maturation, membrane trafficking, GTP loading, and activation. So in absence of RB, NRAS activation is enhanced, inducing DNA damage response activation and cellular senescence. And this functions as a barrier against malignant conversion of adenoma to adenocarcinoma. But one more important question remains whether DNA damage response is really required for the establishment of cellular senescence machinery or not. To answer these questions, we further analyzed RB ATM composite knockout mice and compared to single RB heterozygous mice and ATM knockout mice. You can see here, as I told you in the introduction, these mice generate adenoma or pre-malignant tumor, but additional loss of ATM, these mice generate adenocarcinoma arising from the C cells based on calcitonin expression and carcinogen antigen and proliferation marker Ki67. Importantly, immunohistochemical analysis of these tumors showed a very intimate correlation between DNMT1, DNA methyltransferase, and P16 INK4A expression. And this pattern was a reverse or opposite to their expression pattern in adenoma. This suggests that, or indicate that, germline inactivation of RB and ATM induced malignant transformation in mice, in C cells of mice, and suggests that RB, ATM, DNMT1 might functionally interact to regulate cancer progression. To investigate this hypothesis more, we generated myths of various genotypes of RB and ATM, and 3T3 protocol analysis of these cells showed that RB ATM double knockout myth reached immortal states masked significantly earlier than RB single knockout or ATM single knockout myth. Furthermore, colony formation assay, these cells, RB ATM double knockout myth, they form colonies when they are plated under low seeding condition. Immunoplotting also, analysis of this myth showed they express very high level of DNMT1 and undetectable level of P16, the reverse pattern compared to single RB knockout myth. Also, we assessed the role of ATM directly in myth immortalization, and by knocking down ATM using specific shRNA in RB negative background and RB positive background. Only in RB negative background, ATM knockdown upregulated the chromatin modifiers, particularly the DNMT1, and suppressed P16. So we thought, maybe DNMT1 is a mediator for RB-ATM interaction. So to assess the role of DNMT1, we knocked down DNMT1, and knockdown of DNMT1 induced P16 and suppressed the immortal states of RB-ATM double knockout myth. So here, conclusion of these experiments says that RB-ATM double knockout myth escaped serosinisin, and RB-ATM DNMT1 pathway regulates myth immortalization. Further, we reconstituted ATM to RB-ATM double knockout myth, and this ATM reconstitution, but not ATM phosphorylation deficient mutant, suppressed the colony formation assay. So this means that they are dependent on ATM activation. Immunoplotting of these complemented cells showed that ATM suppressed DNMT1 induced P16, but this was not the case for DNMT3B, which is responsible for DNA methylation, durable DNA methylation. But we asked the question, how RB-ATM interaction regulate P16? What is the mechanism of their regulation of P16? First, we checked or tested the effect of ATM on INK4A transcription. We found that inactivation of ATM by shRNA or a specific kinase inhibitor or germline inactivation suppress INK4A transcription only in RB negative cells, not in RB positive cells. Because INK4A promoter methylation is a common mechanism for INK4A gene regulation, we checked the methylation state of INK4A promoter using bisulfite sequence. The principle of bisulfite sequence is that conventional methods for sequencing methods does not differentiate between methylated cytosine and unmethylated cytosine. However, when the DNA is treated with bisulfite, methylated cytosine stay as cytosine. However, non-methylated cytosine is converted to uracil. So following sequencing of the converted DNA, methylated cytosine stay as C, but uracil can read as T. So comparing these two sequences, we can identify the methylated cytosine from unmethylated cytosine. We performed this bisulfite sequence on wild-type MEF, RB single knockout MEF, and RB ATM double knockout MEF. You can see here an activation of RB suppressed the frequency of methylated CBG in INK4A promoter. However, loss of both RB and ATM very much increased the methylation level in INK4A promoter. Further, we also analyzed RB-ATM double knockout MEF reconstituted with ATM and found that ATM reconstitution significantly suppressed the methylation level of INK4A promoter, but this was not the case with ATM phosphorylation deficient mutant complemented MEF. Importantly, treatment of ATM reconstituted MEF with ATM-specific kinase inhibitor reversed this hypomethylation phenotype. And more importantly, reconstitution of RB after ATM, RB antagonized ATM function in reducing the methylation level in INK4A. So this means that RB antagonized ATM in a similar way to specific ATM kinase inhibitor. Further, we performed CHIP assay using a specific antibody against DNMT1 H3K4 dimethylation, H3K9 trimethylation, and found that RB-ATM deficiency enhanced the binding of DNMT1 and this histone mark to the promoter of INK4A gene compared to RB single knockout MEF. And importantly, when we reconstituted ATM reconstitution suppressed the binding of DNMT1, particularly in the regions closer to the transcription start site. We confirmed these results using a quantitative CHIP assay, and as you can see here, ATM reconstitution significantly suppressed DNMT1 binding to INK4A promoter. This indicates that RB-ATM DNMT1 pathway regulate INK4A promoter methylation. But how RB-ATM interaction regulates DNMT1 itself? First, we check the effect of ATM on DNMT1 transcription. ATM does not affect DNMT1 at transcription level. We went further down and analyzed DNMT1, our ATM status effect on DNMT1 at post-transcription level. We treated cells with proteasome inhibitor MG132. You can see here this is RB-ATM double knockout complemented with ATM. This is ATM phosphorylated at serine 1981. This is a phosphorylation site usually used or commonly used to assess ATM activation. And the green is DNMT1. DNMT1 in RB negative cells is very low, and proteasome inhibitor treatment increased or risked the DNMT1 and enhanced its co-localization with ATM. So this means that ATM regulates DNMT1 at post-transcription level when RB is So what the mechanism, how the molecules or the protein interact together to regulate DNMT1 stability. During this work, one science paper appeared showing that DNMT1 acetylation decrease the protein stability. We checked this in our system and found that, as you see here, this is RB-ATM double knockout myth reconstituted vector or ATM. You can see ATM reconstituted cells has very low level of DNMT1, but MG132 treatment rescued DNMT1. However, most or almost all DNMT1 protein is acetylated and polyapictinated. This means that ATM, in absence of RB, enhance acetylation and polyapictination of DNMT1. We further performed co-immunoprecipitation assay using specific antibodies against DNMT1 and ATM, and found that ATM bound to DNMT1 in a complex with TIP60 and UHRF1. These results indicate that in absence of RB, ATM physically binds to DNMT1 and promotes its acetylation by TIP60 and subsequent apictination by UHRF1. UHRF1 is E3 ligase. So we directly assessed the role of TIP60 in this process and knocked down TIP60 and found that depletion of TIP60 suppressed acetylation of DNMT1 and rescued DNMT1. And also, as reported before, TIP60 depletion reduced activation of ATM. However, overexpression of TIP60 in RB-ATM double knockout myth did not affect the acetylation or the abundance of DNMT1. This means that ATM is required for TIP60 acetylation or degradation of DNMT1. So here is the conclusion. TIP60 is required for ATM activation here. TIP60 acetylate DNMT1 and decrease its stability in RB deficient cells or MEF. ATM is required for TIP60 induced acetylation of DNMT1 in RB deficiency. So ATM is required for TIP60 acetylation of DNMT1. Further, we mapped ATM for the region interacting or binding to DNMT1 by generating these ATM mutants, and found that when we perform immunoprecipitation, we found that the regions which has kinase domain of ATM, this called ESKDKS fragments, only these fragments which contain the kinase domain, they are able to bind to DNMT1 and acetylate, promote its acetylation and degradation in a similar manner to wild type ATM. In a similar way, we mapped DNMT1 for the binding region with ATM by generating these DNMT1 mutants, and found that the region which has the catalytic motifs number 9 and 10 that are able to bind to ATM here, immunoprecipitation. So we think that in this interaction, TIP60, when ATM is bound to DNMT1 through kinase catalytic domain binding, TIP60 acetylate ATM and further enhance its acetylation or autophosphorylation and then activation. At the same time, TIP60 acetylate transferase acetylate DNMT1 and promote its apictination by UHRF1. So this complex, I call it DNMT1 degradation complex. In an aim to investigate the function of the RB-ATM DNMT1 pathway genome-wide, we performed microarray analysis in 2 experiments. The first experiment by directly depletion of DNMT1 using specific shRNA, or indirectly inactivation of DNMT1 by reconstitution of ATM to RB-ATM double knockout method. If you remember, ATM degrade DNMT1. So this experiment should be equal to this experiment. And we found that hundreds of genes are upregulated in these 2 experiments, and about 318 genes are overlapping in these 2 experiments. We further analyzed 3 representative genes of this list, which has a high score, and we found that this is FOX06 and SHIP2 NUC. You can see here ATM reconstitution suppressed the frequency of methylated CBG dinucleotides in these gene promoters in a very much similar way to INK4A promoter. This indicates that this RB-ATM DNMT1 might has a genome-wide function not limited to INK4A promoter. DNMT1 pathway might regulate DNA methylation profile genome-wide. In an effort to find the significant clinical significance of our study, we analyzed human cancer, including human sporadic medullary thyroid carcinoma. These human thyroid tumors, this also rise from C cells and equivalent to the tumors generated in our mice model. And we found that ATM, DNMT1, P16 expression are tightly correlated in RB negative tumors. We also analyzed another group of human cancers. This is esophageal squamous cell carcinoma. And we found that this ATM, DNMT1, P16 expression are also tightly regulated in this cancer. Although the pattern is different, in MTC they lost ATM, but in esophageal cancer they still have functional ATM. But we think that this is very important because, as I tell you later, how this can be regulated epigenetically, tumor suppressor genes and oncogenes. So in conclusion, in wild type cells, it is very well known that RB suppress DNA double strand breaks and thus suppress the ATM activation. In this study, we found additionally that RB also has another mechanism to regulate ATM activation through regulation of DIP60-induced acetylation of ATM. And we think that RB status is important for DNMT1 stability by binding with HDAC. RB HDAC DNMT1 complex make DNMT1 stable and occupy or methylate the promoter of epigenetically regulated genes. And this might be dependent on RB status. And now we are doing some work to connect this to the cell cycle phases. Importantly, the finding in our study that I talked about today, that RB loss enhance ATM activation, and activated ATM bind to DNMT1 in a complex with DIP60. DIP60 acetylate DNMT1 followed by apictination by UHRF1, resulting in degradation of DNMT1 and freeing the promoter of target genes, and they are expressed. Here, important notice here, HDAC is out of this complex. So HDAC should stabilize DNMT1. It is out of this complex. However, In case of loss of both RB and ATM, each DAC is bound to DNMT1. DNMT1 is stable methylate, the promoter of epigenetically regulated genes. And importantly, we think that this RB-ATM DNMT1 interaction regulate a very important epigenetic barrier between pre-malignant and malignant state. So this epigenetic barrier includes tumor suppressor genes. We gave an example today of tumor methylation, but however, the esophageal squamous cell carcinoma showed another pattern of methylation or expression pattern of these proteins. So we think also oncogene might be involved in this barrier. So freeing this oncogene or promoter of this oncogene might also contribute to malignant transformation as well as suppression of tumor suppressor gene. This is our collaborators. We thank Professor Noda and Professor Makoto Takito Sensei and Dr. Igra. And also we would like to thank Professor Doki in Osaka University for providing human samples, such as squamous cell carcinoma, Tokyo Women's University, Professor Okamoto for providing MTC samples, Cancer Institutes, Professor Morton and Ijihara, Harvard Medical School, McQueen, and Ronald Dubino for mice, MIT Tarjax for mice, and ST Julie Children's Research Hospital, Macon for ATM mice, and University of Texas Southern Medical Center, David Chen for the expression, ATM expression vectors, our Cancer Research Institute, and our laboratory group. Thank you very much, and I'll be happy to